RSI Poultry Veterinary Consulting LLC presents the analysis of Salmonella Infantis whole genome sequencing data generated from isolates recovered from comminuted chicken samples collected in Delaware. The purpose of this study was to use the analysis of Salmonella Infantis whole genome sequencing data to support Salmonella control strategies in poultry. This video presentation has 4 sections that include the description of the samples, the tools and methods used in the analysis, the results, and the conclusions and recommendations. The genetic data of 78 Salmonella Infantis isolates were evaluated. The isolates were recovered from comminuted chicken samples collected by the FSIS in Delaware between the years 2017 and 2021. The genetic data were downloaded from the NCBI website on August 3, 2021. The references used for the genetic comparisons include a Salmonella Infantis isolate recovered from chickens in the US in 2015, which was one of the first isolates reported as containing the plasmid of the emergent Salmonella Infantis clone in the US. We also include as references 7 Latin American strains, 1 isolate from Europe, and 1 US isolate that did not carry the plasmid of the emergent Salmonella Infantis clone. The Genius Prime software was used to build the single nucleotide polymorphism or SNP phylogenetic tree. It was also used to identify SNP carrying genes or genes carrying mutations, and also to evaluate the plasmid gene content. The Center for Genomic Epidemiology online resources were used to detect the plasmids and to predict the antibiotic resistance profiles. The rapid annotation using subsystem technology online tool, RAST, was used to identify genes associated to various Salmonella cell functions. In addition to the analysis mentioned, we ran statistics to compare the antibiotic resistance profiles. We utilized the Mann-Whitney U-test and logistic regression. Phylogeny results. This is the single nucleotide polymorphism phylogenetic tree. The chromosome of the Salmonella isolates from comminuted chicken, represented here color code based on the year of isolation, were compared towards the US reference strain from 2015, represented here as the reference US reference 1. The references from Latin America in this group, references 2, and also here, references 3, and the European reference also falling in this group of references 2, and the US reference without the plasmid of the Salmonella Infantis emerging clone were all also compared towards the US reference 1. What we determined was that all the isolates from comminuted chicken were highly related at the genetic level, and they cluster in 2 phylogenetic groups, Group A and Group B. And these 2 groups match the SNP clusters reported by the FSIS for these samples. And Group A was more closely related to the US 2015 reference, and Group B was more closely related to the reference without the plasmid of the emergent Salmonella Infantis clone. Plasmid identification results. 2 plasmids were detected. The plasmid of the emergent Salmonella Infantis clone, also named incompatibility F1b plasmid, was detected in 97% of the isolates. And an incompatibility X1 plasmid was detected in 1 isolate, which also carried the plasmid of the emergent Salmonella Infantis clone. The 2 remaining isolates did not carry any plasmid. Overall, all the isolates carrying plasmids were located in the phylogenetic group A, and the isolates that did not contain any plasmid were located in the phylogenetic group B. Antimicrobial resistance results. All the Salmonella Infantis that carry the plasmid of the emergent Salmonella Infantis clone had a chromosomal point mutation in the GRA gene, and this mutation provides resistance to nalidixic acid and ciprofloxacin. All the isolates were also resistant to the aminoglycosides tobramycin, amikacin, and streptomycin. 93% of the isolates were resistant to tetracycline and sulfamethoxazole. 60% of the isolates were resistant to gentamicin. 53% of the isolates were resistant to chloramphenicol. 40% of the isolates were resistant to trimethoprim. 39% of the isolates were resistant to beta-lactam antibiotics, and this resistance is associated to the presence of the BLAST-TXM65 gene in the plasmid, and 19% of the isolates were resistant to fosfomycin. We subdivided the group A into groups A1 and A2 to investigate if any difference in antimicrobial resistance is linked to the location of the isolate in the phylogenetic tree, and if the presence of the BLAST-TXM65 gene can be linked to resistance to non-beta-lactam antibiotics such as chloramphenicol, trimethoprim, gentamicin, or fosfomycin. Regarding the phylogenetic group B, this group is formed by those isolates that do not carry any plasmid or any antibiotic resistance. This chart shows the comparison between the groups A1 and A2 based on the number of isolates that show resistance to the antibiotics or had presence of the BLAST-TXM65 gene or the QACE delta 1 gene that provides tolerance to quaternary ammonium compounds. We determined that the group A2 had more isolates carrying resistance to beta-lactam antibiotics and also to the aminoglycoside antibiotic gentamicin. Also, this group had more isolates containing the BLAST-TXM65 gene. We also determined that the presence of the BLAST-TXM65 gene can be linked to resistance to trimethoprim, chloramphenicol, gentamicin, and fosfomycin. And fosfomycin resistance can be a good predictor of the presence of the BLAST-TXM65 gene. In other words, Salmonella Infantis carrying resistance to fosfomycin can potentially carry the BLAST-TXM65 gene and resistance to beta-lactam antibiotics. Gene content results, plasmid and chromosome. Let's start with the plasmid analysis. Overall, a total of 41% of genes in the plasmid were shared between all the isolates carrying the plasmid. Among those genes identified, we have the K88 fimbria, which is a group of genes that participate in host cell attachment. We have the CCDA and CCDB genes, which are involved in plasmid maintenance in bacterial populations and have been suggested as potential drug targets. Type 1 fimbria, which is involved in host cell addition and BAF formation. The streptomycin 3' kinase gene that participates in the inactivation of streptomycin. The Yersinia bactin genes, which are a highly efficient iron acquisition system. The expression of these genes is induced by iron limiting conditions, and it has also been linked to resistance to copper toxicity. The ADIA and the ADIC genes are responsible for acid resistance. The HHA gene affects biofilm formation by repressing type 1 fimbria, and the TEHA gene provides telluride resistance, and the overexpression of this gene may cause hypersensitivity or resistance to certain disinfectants or antiseptics. A total of 59% of genes in the plasmid differ between the isolates. This chart shows those variables genes identified in the plasmids. There is a group here of genes that are present in more than 90% of the isolates. But also, there is a group of genes that are present in less than 86% of the isolates. If we take a look at those genes that were present in less than 86% of the Salmonella Infantis isolates over the years between 2017 and 2020, we notice that there is a tendency to a reduced frequency of isolates containing genes that provide resistance to arsenicals, beta-lactam antibiotics, fosfomycin, chloramphenicol, trimethoprim, and some aminoglycosides. One Salmonella Infantis carried an incompatibility X1 plasmid. This plasmid lacked the BLA, MRK, and the OQX genes, which are associated to beta-lactam resistance, Fimbria biogenesis, and quinolone resistance, respectively. However, this plasmid kept the PIL-X genes, and the PIL-X genes produce type 4 Fimbria. Type 4 Fimbria has been associated to horizontal gene transfer and may participate in the stabilization of biofilm and has been suggested as a potential vaccine target. Now we move to the analysis of the chromosome gene content. Here, we will roughly compare the SNP carrying genes or genes carrying mutations in the phylogenetic groups 1, 2, and group B. We further subdivided the groups A1 and A2 into alpha and beta based on the resistance to antibiotics. The alpha groups A1 and A2 contained isolates that show resistance to 7 antibiotics. And the beta A1 and A2 groups contained isolates that show resistance to more than 13 antibiotics. With this subdivision, now we move into the analysis of genes carrying mutations, as we will see in the next slides. The first step into the analysis was to locate the areas in the chromosome where most of the mutations are located for each of the groups. We observed that the alpha, beta, A1, and A2 groups had more mutations beyond the 0.4 megabase nucleotide position in the Salmonella chromosome. And the phylogenetic group B had most of the mutations located before the 58 kb nucleotide position in the Salmonella chromosome. And in this region, the group B had mutations located in phage, recombination, and one amino acid biosynthesis gene. The next region is located between the 0.4 and the 1 megabase nucleotide position. In this region, the groups A1 and A2 had mutations in 2 genes participating in amino acid metabolism. The next region is around the 1.7 megabase nucleotide position in the Salmonella chromosome. The Salmonella pathogenicity island 1 is located in this region. Here we have that the groups A1 and A2 had mutations in some virulence transported and also one exopolysaccharide synthesis gene. The name of the genes are followed in parentheses by the number of isolates that contain the mutation and the location of the isolate in the phylogenetic tree. I want to highlight here this gene, the MFS. The MFS gene is a major facilitator superfamily gene. This gene has a premature stop codon that suggests this gene is not functional, and a non-functional gene may cause reduced tolerance to certain drugs. The phylogenetic group B had mutations in transcription and replication genes in this region of the chromosome. The next region is located between the 2.5 and the 3.9 megabase nucleotide position in the Salmonella chromosome. In this region, we observed that most of the mutations in the group A isolates are located in one transposase gene. Other mutations were located in biofilm, virulence, Fimbria, and stress response genes. Regarding the group B isolates, Most of the mutations were located in the OADA metabolic gene. Other mutations were located in one acid resistance gene, virulence genes, and one fimbrial biogenesis gene. I would like to focus on this gene, the KDSA gene. This gene functions in lipid A maturation and has lost the stop codon of a normal gene. This mutation may alter gene functionality and the production of a normal LPS. The last region is located beyond the 3.9 megabase nucleotide position in the Salmonella chromosome. The A group isolates had most of the mutations in the acid tolerance genes ADIA and ADIC and two transposase genes. Other mutations were located in a membrane, a transcription factor, a virulence, and a metabolic genes. I want to highlight here this gene, the VTUB gene. This gene had a premature stop codon that may cause a decrease in the production of the VTUB protein, which is known to cause resistance to colicins. In the group B, most of the mutations were found in one transposase gene and one quorum sensing gene. Overall, we conclude that the analysis of genes carrying mutations was useful to distinguish between phylogenetic group A versus phylogenetic group B, and can be relevant to distinguish also specific isolates of interest. Now this slide shows the gene content identified in the whole genome sequencing data, plasmid and chromosome using RAST. This was done in order to detect any gene that may have not been accounted for during the analysis towards the reference genomes. We compare the gene content of the phylogenetic groups A towards the phylogenetic group B. The A1 alpha isolates carry several antimicrobial genes that were not present in the phylogenetic group B. Here, highlighted in yellow, are those genes identified by this approach. The A2 alpha isolates were similar to the A1 alpha isolates, although this group contained additional aminoglycosides resistance genes not present in the phylogenetic group B. The A1 beta isolates carry additional genes associated to resistance to beta-lactam antibiotics, fluorphenicol, and fosfomycin. These isolates also carry genes of the type 4 secretion system. The subgroup A2 beta isolates were very similar in gene content to the A1 beta subgroup. From this analysis, we conclude that the alpha groups resulted more similar to each other than to the beta groups. To sum up, we analyzed genomic data of 78 Salmonella Infantis isolates recovered from comminuted chicken samples collected by the FSIS in Delaware between the years 2017 and 2021. The genetic data was used to determine phylogeny or genetic relationship between the isolates and references, and also to determine the plasmid content, antimicrobial resistance profiles, gene content, as well as mutations in genes that may help differentiate isolates between or within phylogenetic groups. From the phylogeny analysis, the Salmonella Infantis isolates from comminuted chicken were highly related at the genetic level, and they form two phylogenetic groups, which were compatible to the two SNP clusters reported by the FSIS for these samples. We identified the plasmid of the emergent Salmonella Infantis clone in 97% of the samples. Two isolates did not contain any plasmid. The presence of non-plasmid containing Salmonella Infantis in comminuted chicken may indicate the relevance of cross-contamination in processing. The BLAST-CTXM65 gene was linked to more antimicrobial resistance in Salmonella Infantis. The presence of the BLAST-CTXM65 gene was also linked to resistance to trimethoprim, chloramphenicol, gentamicin, and fosfomycin. nalidixic acid and ciprofloxacin resistance was carried in the chromosome. Regarding the plasmid gene content, some genes that provide resistance to antibiotics seem to be less frequent in Salmonella Infantis isolates recovered after 2017. Some plasmid genes were common to all the Salmonella Infantis carrying the plasmid. Genes such as the CCDB, K88 fimbria, or Yersinia Bactin genes can be used to identify Salmonella Infantis carrying the plasmid. The plasmid also carries genes that provide Salmonella Infantis with beta-lactam resistance, UV light, quaternary ammonium compounds, mercury, and arsenic tolerance. These genes are in a group of plasmid genes which are named variable. These variable genes can be used to distinguish isolates. The chromosome gene content analysis was based on the study of the differences in gene mutations. Phylogenetic groups A and B differ in the chromosomal location of gene mutations. In addition, groups A and B may differ in their amino acid metabolism and in the presence of mutations in genes of the Salmonella pathogenicity island 1. We detected mutations affecting stop codons, such as the mutation in the VTUB gene that may provide Salmonella Infantis with resistance to colicins. We conclude that genes with mutations might be useful to distinguish individual isolates of interest. What are some of the recommendations generated from this work? In the first place, Salmonella Infantis that contaminate comminuted chicken might not necessarily carry the plasmid. The plasmid of the emergent Salmonella Infantis clone is known to confer Salmonella Infantis with survivability advantage that may complicate the control. So, if Salmonella Infantis not carrying the plasmid is found in this type of poultry product, a special attention should be put in tightening the control at the processing plant. To learn what type of Salmonella Infantis is circulating in the environment, the detection of some target genes will be helpful. A panel of shared invariable plasmid genes can be created for the detection of the pathogen. An antibiotic resistance gene detection or a Kirby-Bauer or a sensitized AST can be also used to complement the characterization. Further characterization may include the detection of certain relevant mutations that may affect control interventions, such as the mutations in the VTUB gene that may confer the isolates with colicin resistance, which can be relevant when probiotics are used, or mutations in the ADIA acid tolerance gene that may confer survivability advantage in acid treatments. The detection of the Blasity XM65 gene was a very good indicator of high tolerance or resistance to more than 13 antibiotics, including trimethoprim, chloramphenicol, gentamicin, and fosfomycin. Tracking this gene in the environment might be a very good tool to measure control interventions. Therefore, to evaluate the efficacy of control interventions, the detection of Salmonella Infantis should be complemented with the detection of target genes selected based on the specific situation of a poultry integrator, although the detection of target genes may suffice. So what's next? How can we start using the information generated here? One approach to complement the control program for Salmonella Infantis can be the selection of Salmonella Infantis target genes for the design of a PCR-based screening method, and with this tool support an epidemiological study in order to determine prevalence, distribution, or the origin of Salmonella Infantis. The results of this epidemiological study will help the identification of more appropriate control interventions. Once the control intervention strategy is put in place, its effectiveness can be evaluated by the use of this same PCR screening method. Finally, this study has been performed on a specific group of isolates from one region of the United States and one type of poultry product. The findings can be also used to inspect other Salmonella Infantis isolates to study the diversity in other regions or poultry products. These are our references. Thank you very much for taking the time to review this presentation. Please address any questions or comments to the following email address. Thank you.